We are gathered today here this evening to witness the 60th Inaugural Lecture as part of the Inaugural Lecture series in the university. An Inaugural Lecture is a sacred tradition of the university as a rite of passage. It is a moment whereby a scholar reflects back on the work he has done so far up to the time he became a professor in that field. And there are certain fundamental questions which the scholar will attempt to answer during the presentation. And part of that is what contribution has he made to knowledge generally. So today we are witnessing this very beautiful and laudable tradition of the university. Today Professor Muhammad Bashir Bello will be addressing us on a profoundly urgent and fascinating topic, which is confronting viruses with vaccines from hard health to human survival. The global events that have happened in the recent past have taught us that human health is intimately connected and linked with animal health. The invisible battle against viral pathogens is fought daily by brilliant minds in the laboratory, and Professor Muhammad Bashir Bello has done that in his quest for knowledge in laboratories and also on the field. So today, ladies and gentlemen, you should prepare to have your perspectives broadened in that area. Your intellectual curiosity is going to be sparked, and your appreciation for the field of veterinary microbiology is going to be profoundly deepened. Therefore, on behalf of the Vice Chancellor, Professor Bashir Garba, MFR, the Senate, and the entire university community, I formally welcome you to the 60th Inaugural Lecture in the Inaugural Lecture series of the university, and I declare this lecture open. And I hope that we will listen keenly and we will take a lot of things with us as we leave today.
Bashir was born on the 13th April 1984 in Tambuwal local government area of Sokoto State, Nigeria. He began his academic journey at Shehu Shagari College of Education Staff Primary School, Sokoto in 1995, and later proceeded to the famous Nagarta College, Sokoto, where he obtained his West African School Certificate in the year 2001. He obtained his Doctor of Veterinary Medicine degree in 2007 from Usman Danfodio University, Sokoto, the most peaceful university in Nigeria. Following his National Youth Service at the Ekiti State Ministry of Agriculture and Rural Development in 2009, he briefly served the Sokoto State Government between 2009 and 2012 before joining the services of Usman Danfodio University, Sokoto as an assistant lecturer. He steadily rose through the ranks of Lecturer 2 and then Lecturer 1 following the completion of his master's degree program from the prestigious Ahmadu Bello University, Zaria in 2013. His craving for professionalism in advanced technology-driven society was recognized by the University Putra, Malaysia, and was awarded a PhD admission in 2015 where he obtained his PhD degree in immunology and vaccinology in the year 2018. He was then promoted to the rank of senior lecturer in 2021. Throughout his academic journey, he developed enduring interest in infectious diseases, molecular virology, and vaccine research. He attained the highest rank of professor effective 1st January 2026. Professor Muhammad Bashir Bello is a professor of veterinary virology at Usman Danfodio University, Sokoto, Nigeria, and currently serves as an associate research scientist in the Vaccine Development Division, Infectious Disease Research Department, King Abdullah International Medical Research Center in Riyadh, Saudi Arabia. He is a molecular virologist and vaccinologist whose research integrates genomic surveillance, molecular virology, immunology, bioinformatics, and vaccine development to address emerging and re-emerging infectious diseases within the One Health framework. Mr. Vice Chancellor, sir, over the years, Professor Bello's research evolved from livestock and avian virology into broader areas of genomic surveillance, modern vaccine development, and translational infectious disease research. During the COVID-19 pandemic, he became actively involved in national preparedness and response activities in Nigeria. He contributed to COVID-19 diagnostic effort in Sokoto, became a member of the Nigeria COVID-19 Research Coalition under the Nigeria Center for Disease Control, and later served on the Technical Advisory Committee of the Nigeria Governors Forum on COVID-19, contributing to discussions on vaccine research, preparedness, and development during the pandemic. Professor Bello was also among the scientists involved in establishing the National Vaccine Consortium of Nigeria, where he served as technical lead in efforts aimed at strengthening local vaccine research and development capacity. Through this initiative, the consortium secured a TED-fund mega grant research grant that supported the development of one of Nigeria's earliest candidate DNA vaccine against the severe acute respiratory syndrome coronavirus 2. He previously served as Senior Research Associate and Pioneer Deputy Director Research and Services at the Center for Advanced Medical Research and Training, CAMRET, here in Usman Danfodio University, Sokoto, where he led important projects on Lassa fever and COVID-19 vaccines. Mr. Vice Chancellor, sir, Professor Bello subsequently moved to King Abdullah International Medical Research Center in Riyadh, where he is actively involved in genomic surveillance and messenger RNA vaccine development against emerging and re-emerging viral diseases. He currently leads and contributes to several advanced vaccine research projects targeting pathogens of regional and global public health importance, including the development of next-generation messenger RNA vaccines and a flexible virus-like particle vaccine platform aimed at improving rapid response capacity against future emerging viral threats. He has secured several competitive research grants as principal investigator, including grants from the Tertiary Education Trust Fund, the International Foundation for Science, and the Saudi International Institute of Health. In addition to his research activities, Professor Bello remains deeply committed to teaching, mentorship, and scientific capacity development. He has supervised and mentored numerous MSc and PhD students to completion, while many others are currently undergoing training under his supervision in vaccinology, molecular virology, bioinformatics, and infectious disease research. He has taught courses in virology, immunology, molecular biology, vaccinology, and bioinformatics at both undergraduate and postgraduate levels. His commitment to scientific capacity building in Africa is further reflected in his role as a visiting professor at the East African Center of Excellence for Vaccines, Immunization, and Health Supply Chain Management from the University of Rwanda, where he contributes to postgraduate training and vaccinology capacity development across the continent. He is also a visiting professor at the Center for Vaccine Research and Biotechnology of the Federal University Lafayette Teaching Hospital in Nigeria. Distinguished ladies and gentlemen, Professor Bello's scientific contributions are reflected in more than 100 peer-reviewed publications, numerous invited lectures, conference presentations, collaborative research projects, and competitive grants across multiple international institutions. His work has appeared in leading international journals, including The Lancet, Nature Medicine, The Lancet Infectious Diseases, and the Journal of Infection, among others. According to the Google Scholar, Professor Bello has accumulated more than 9,000 citations and has an H-index of 27, reflecting the impact and recognition of his scholarly contributions within the global scientific community. In addition, Mr. Vice Chancellor, sir, Professor Bello serves as a postgraduate thesis examination at Ahmadu Bello University, Zaria, and King Saud bin Abdul Aziz University for Health Sciences in Riyadh, Saudi Arabia. He is an active member of several professional bodies, including the International Veterinary Vaccinology Network, the World Society for Virology, the African Society for Virology, the Biotechnology Society of Nigeria, and the Nigeria Society for Immunology. Throughout his career, Professor Bello has consistently advocated for stronger genomic surveillance system, local vaccine manufacturing capacity, biotechnology innovations, and preparedness-driven approaches to infectious disease control. His scientific philosophy is firmly rooted in the One Health concept, recognizing the interconnectedness of animal health, human health, and environmental systems in addressing emerging infectious diseases. Professor Muhammad Bello Bashir is married and blessed with 4 children, 3 boys and 1 girl. He currently resides in Riyadh, Saudi Arabia. Mr. Vice Chancellor, sir, thank you very much for giving me this great opportunity to read this citation.
Innovation and Development, Academic and Administration, the Registrar, members of University Governing Council and the Senate, Provost Postgraduate School, Provost College of Health Sciences, deans of various faculties here present, especially that of veterinary medicine where I come from, professors, directors of various research institutes and university research centers, heads of department here present, particularly that of veterinary microbiology, my very good friend, my teachers, mentors, mentees, friends, family, collaborators, I greet you with the best greetings in Islam. Assalamu alaikum wa rahmatullahi wa barakatuh. I feel greatly honored, Mr. Chairman, today, not just because I'm going to be presenting my inaugural lecture as a professor in this university, but also because I am doing so in the midst of 2 most important people in my life. And these are my parents, Dr. Bello Muhammad TAMBUWAL, mashallah, still looking young, smart, energetic, and charismatic. And indeed, my beloved mom here, Hajia Fatima Bello TAMBUWAL, whose prayers, efforts, encouragement, and support have, subhanallah, been the reason why I have succeeded in life. God knows how many times in my life I went to Masjid al-Haram in the Kingdom of Saudi Arabia and prayed to God Almighty to grant me such an opportunity to witness this day alongside with them. And all I will say, Mr. Chair, is Alhamdulillah, Alhamdulillah, alladhi bi ni'matihi tatimmu salihat. May Allah Almighty continue to grant us wisdom, energy, and capacity to put smiles onto the faces of our parents. Having said this, Mr. Chairman, quickly, I'll be presenting my inaugural lecture today. And the title of this lecture is Confronting Viruses with Vaccines from Herd Health to Human Survival. Mr. Chairman, sir, this presentation is not merely recounting my scientific experience as a virologist or as an expert in the field of vaccinology. No, it is about reflecting on the entire trajectory of my academic and professional journey. When I look through the years, Mr. Chairman, I realize that my journey is full of transitions. It has been a journey from animal health to global health, from basic science to translational research, from field epidemiology to vaccine development, and from local veterinary clinical practice to global and international scientific engagement. Mr. Chairman, I'm a trained veterinarian. I started my career as a field veterinary officer, working very closely with farmers and herders, especially Fulani herdsmen, in some of the rural settings here in Nigeria. But subsequently, I transitioned first into university system, and subsequently I found myself in one of the topmost medical research institutes in the world, the King Abdullah International Medical Research Institute, Riyadh, Saudi Arabia. Not as a veterinarian, but this time around as a full-fledged research scientist, working on all aspects of viral biology and vaccine development against emerging and re-emerging infectious diseases of huge global health concerns. But Mr. Chairman, sir, I am so glad that despite all this, I remain focused. In fact, I realized that throughout my professional life, I have been doing one thing and one simple thing: pursuing solutions for viral threats of both human beings and animals. And that is why I entitled this presentation, Confronting Viruses with Vaccines from Herd Health to Human Survival. Mr. Chairman, sir, all these transitions, I have to admit, did not occur in any way by my plan. Neither did they occur as a result of any smartness on my own side, but they occurred as a result of some unexpected opportunities, such as the emergence of COVID-19 pandemic, but more importantly, by my scientific curiosity and my willingness to accept challenges at different phases of my lifetime. And that is why in this presentation, Mr. Chairman, I will be highlighting my early experience in veterinary science and how I, for the very first time, fell in love with the field of vaccinology and virology, and how I subsequently found myself doing what I enjoy doing the most: designing and developing vaccines against emerging and re-emergent viral threats, not only in Nigeria or in Africa, but indeed in the Middle East, especially in the Kingdom of Saudi Arabia, where I currently reside. Mr. Chairman, sir, if there's anything this journey has taught me so far, it is that I have stopped seeing diseases as single, independent biological entities. No, I now see diseases as complex challenges that devastate the world, that tend to affect food security, global health security, and substantially retard national development. Ladies and gentlemen, I want to use this opportunity to once again welcome you all to my inaugural lecture entitled Confronting Viruses with Vaccines from Herd Health to Human Survival. You are moving a bit faster then. Mr. Chairman, sir, my journey starts in a very modest town called Tambuwal. Tambuwal is located, for those of you who don't know, in the extreme northwestern part of Nigeria, and I hope people from Gidadawa will not blame me if I told them it's the best local government in Nigeria. Mr. Chairman, in Tambuwal local government, we don't anymore count the number of professors we have, not even per given district. But because in Kaswagari area where I come from, Mr. Chairman, I am the 6th or even the 7th professor. The first being the former Vice Chancellor here, Professor Rizkwa Arabushehu, followed by my beloved uncle, Professor Farouk Muhammad Tambol. So even within my family, Mr. Chairman, I am not the first professor. So this is to tell you how we take education in my own family and in my own locality. I still remember when we were quite young, and how my dad here used to tell us that you just have to study, because the surest way to success lies between paper and pen. And I could still hear his words through the vicissitudes of time, quoting this popular quote. He used to tell us that tarumul izza summa tanamu laylan yagusul bahra man talaba al-ayy. Mr. Chairman, he used to say that you want to be great in life and yet you sleep throughout the night. He who looks for diamonds had to dive deep down into waters. Mr. Chairman, at that time, these words did not make a lot of sense to me. But much later, I was to study this book, Ta'alimul Muta'allim, and I came across this particular verse. Then the message became much clearer. But the information had already been engraved and imprinted in my medulla oblongata, so I already have the message. Later, I just appreciated its meaning. Mr. Chairman, this was my childhood. Even before I attended my primary school, I started my primary school at Sheikh Shagari College of Education, Primary School, Sokoto, where, alhamdulillah, I finished and graduated as the best student in my class, obtaining 5 out of 9 prizes during that unique speech and prize giving day held under the chairmanship of late Dr. Abubakar Muhammad Sokoto. May Allah forgive him. I then moved to Nagata College, Sokoto, where I consistently was also among the top students, and I subsequently became the head of students in the year 2001. And Mr. Chairman, sir, not only did I become the head of student in the year 2001, I graduated also with the best SSCE result with distinctions and credits in all the subjects I sat for in that particular year. It was also in Nagata, Mr. Chairman, that we began to appreciate the values of life. I still remember what our teachers used to tell us, particularly our principal, the former principal. And he used to tell us, there is no shortcut to success. That success, anywhere you see shortcuts, danger lies ahead. And behold, we stick to those words. And I tell you, he was telling us that if you do not make requirement to go to university, don't even greet me when you see me next time. And 2 years after our graduation, more than 70% of our class members made it to university. Thank you, Baba Umaru, for those words of wisdom. It was also at Nagata College, Mr. Chairman, that I came to be known by a nickname, Calculo, or some people used to call me Calculator, because of my extreme passion for mathematics. Mr. Chairman, I was so involved and in love with mathematics. By the way, the name Calculo, I inherited it from my senior, who is incidentally a professor here, Professor M.S. Abubakar, my senior at Nagata. And now we are also colleagues in the same faculty. So this was my life, Mr. Chairman, at Nagata College, Sokoto. And I was naturally disposed to study anything that was inclined with mathematics. But then my only ambition was to become an engineer. And this was what I planned for. But then, subsequently, as fate would have it, Osman Onfodi University, Sokoto, in those days, did not offer engineering. My friend convinced me to go for human medicine. Subsequently, I was admitted into the School of Veterinary Medicine. When I was admitted into vet medicine, Mr. Chairman, initially I hesitated, but some voices stood out. One of them was that of my father. He told me, go and study veterinary medicine. Don't you see your Uncle Farouk? He was referring to FM Tambuwal. Didn't you see Abdul Qadir? He was referring to Prof. AUZ-CINEMATOGRAPHY. These are people that have made their names and written their names with golden pen, not only in the university, but also in the veterinary profession. The other voice, Mr. Chairman, was that of my friend, Mubarak BELLO. I remember how Dr. Mubarak used to visit me on his bicycle, Mr. Chairman, at my own house. He was, by the way, also admitted into the same veterinary medicine together with me. He tried to convince me, and subsequently I accepted. Mr. Chairman, sir, I feel proud to mention it now. Accepting to study veterinary medicine is one of the most rewarding decisions that I have ever made in my life. Thank you, Baba, and thank you, Mubarak, for such advice. So, Mr. Chairman, this was me at university. So I accepted to study veterinary medicine. I was in School of Vet. Everybody knows how rigorous veterinary school is. If you escape the tight junctions of anatomy for Prof. Somfada, you will not cross the oceans of pathology for Prof. Tambol. And if you also escape all this, Prof. Ade, just scattered blood is waiting for you to lacerate you in surgery and radio. But in spite of all this, Mr. Chairman, alhamdulillah, I finished scot-free. I was among the very few privileged ones to have graduated without having ever researched any course throughout my life. Thank you. I mean, I really appreciate Allah for all this, Nema. Okay. So, this was me And then it was also, Mr. Chairman, during this session that I first came across the field that will ultimately define my career. And this is virology. And the source of inspiration for that particular course that made me what I am today is this right gentleman standing, sitting beside Prof. F.M. TAMBUWAL, Professor BELLO Rabiu Alkali. Prof. Alkali, Mr. Chairman, he has been the source of inspiration. He is my first mentor in the field of virology. When I saw him, I see virus. And in fact, so much was I fascinated with virology that my classmates began to associate me with the name BBT virus because of the passion I have for virology. This passion grew until I graduated and then moved to ABU Zaria. I still pursued my degree in the field of virology under the tutelage of another professor, Professor H.M. Kazim, whom I was opportune to learn a lot of things, such as diagnostic virology, clinical immunology, viral disease dynamics, and so on. It was also Prof. Kazim that exposed me to the aspects of critical scientific thinking. All this, I thank him for all that. I was also opportune to pass through Professor Junaid Kabir, who exposed me deeply to viral epidemiology at such an advanced level. This prepared me, Mr. Chairman, to go for PhD, which is where I can say I was made as a scientist. In my PhD, Mr. Chairman, I met Prof. Rahman as my supervisor. Prof. Rahman, I still remember what he told me the very first day he saw me. He said, Bashir, if you come to me, if you walk to me, I will run to you, he said. That if you are ready to do the work, I will be ready to assist you. It seemed as if we were doing the PhD together. Mr. Chairman, it was in his lab that I learned how to dissect a virus and reassemble it, Mr. Chairman, through a technology known as reverse genetics. I also learned a lot of things that have to do with rational vaccine design, that have to do with translational vaccinology, infectious diseases, and so on. Okay, sir. Yes. So, Mr. Chairman, sir, this is my educational career. So before we now go to, can you go to, before we start presenting the exact content of the lecture, Mr. Chairman, it is important to mention some of the key terms that will define my career, that will be instrumental to understanding what I will be presenting. And these are three things. We have virus and molecular biology. For those of you who do not appreciate what a virus is a very tiny entity. It is so tiny, Mr. Chairman, that you have to assemble around 10,000 pieces of coronavirus to form a diameter of a hair strand. Virus is so tiny, yet the consequences are severe. The consequences of virus, even every damn pandemic that has occurred in the history, perhaps with the exception of cholera and maybe Black Death, every damn pandemic that has occurred is as a result of a virus. Whether it was smallpox that occurred more than 700 years ago, or Spanish flu 1918, that killed more than 50 million, or HIV pandemic that started 1980s and still ongoing, or even the SARS-CoV of 2002, MERS of 2012, and even the mighty COVID-19 that engulfed the life of so many people and brought the world to stagnation. So viruses are tiny, but they are quite significant. Viruses are also said to be simple in structure, but they are highly diverse. Simple in the sense that they are made up of only protein and nucleic acid, but then they are quite diverse because they can be DNA or RNA. They can be segmented or non-segmented. They can be single-stranded or double-stranded. They can be positive sense or negative sense. So they also have different diversity. Here comes the question again. Are these viruses alive or dead? Many people believe that viruses are dead because, as my teacher used to say, they are metabolically lazy pathogens. They are metabolically lazy because they lack all the necessary machineries necessary for them to perpetuate on their own independently, but they are able to replicate and cause disease once they find themselves in the living host. So that is the dilemma, and it's still ongoing, the debate whether the viruses are still alive or they are not living. The other component that will define my work is vaccines, Mr. Chairman. So if viruses are said to be missiles that move in the airspace of any country. For instance, vaccines can be said to be the interceptors of these missiles. Vaccines, in other words, are things, a whole or partial portion of the organism that are able to induce immune response but unable to cause any disease. There are so many types of vaccines, so many platforms. There is live attenuated kill vaccines, and of course next generation vaccines such as messenger RNA, recombinant DNA, and so on and so forth. Lastly, we have molecular biology. Everything I will subsequently discuss will depend on molecular biology. Molecular biology is like the language of modern virology, because the key to understanding molecular biology is what we call central dogma, which shows flow of genetic information from DNA to RNA and RNA to proteins. Every function in the body is performed by proteins, but the information for the production of that protein is engraved in our DNA through an intermediate known as RNA. So this, through molecular biology, you will be able to study disease pathogenesis, study viral epidemiology, design new vaccines, and trace outbreaks in real time. So, Mr. Chairman, quickly now I will start my research contributions in the field of livestock virology. Next. Every veterinarian, Mr. Chairman, knows the value of PPR. PPR is a disease that devastates flock of sheep and goats. And we know the importance of sheep and goats. Sheep and goats are very important, not only as suppliers of animal protein for human survival, not only as suppliers of also hides and skin for other commercial purposes, but they are also, in fact, they are seen as poor man's ATM machine, because they are used in emergencies for solving some immediate problems. Yet we have a disease called PPR that devastates them, so that when it occurs in a flock, more than 90% of the flock can go away as a result of this disease. Because of the menace of this disease, Mr. Chairman, Foods and Agricultural Organization, as well as Organization for International Epizootics, they came up with a plan to eradicate this disease. Nigerian government, for instance, federal government and state government, through the state government, they are annually vaccinating against this disease, but yet the prevalence of this disease is still increasing. So this was the real thing that motivated me to move into the work when I was in ABU's area. I was in ABU, I tried to investigate what exactly is the real situation on ground about this disease in our own locality. And believe you me, the finding was amazing, because we tried to sample only unvaccinated sheep and goats in our locality, random sampling, yet we found that 1 out of every 2 sheep and goats were infected with this virus, meaning that the virus is actively circulating in our own environment despite annual vaccination. And even if, which means if we keep vaccinating the way, at the rate we were doing, we will never control this disease. So we recommended that there is need to intensify the vaccination. Because according to certain philosophy, intensification of vaccination up to the level of 80% herd immunity is what is required to eliminate this disease. But we were in nowhere near that. So this was the first recommendation we did about our work when we did our MSc. Now, as a follow-up to this study, Mr. Chairman, we had a PhD student, a very intelligent PhD student, Dr. Zaid Shehu, who happens to be a staff also in this university. Mr. Chairman, building on this work, we asked Dr. Zaid to go and invest, since we established the presence of this virus. So we asked Dr. Zaid to go and find what exactly is the genetic makeup of this virus in this our locality. And behold, he did it. We detected this virus by molecular means, and not only detected it, we characterized it and obtained that when we showed from the phylogenetic tree that the virus we have in our locality is what we call lineage 2, and we reported that this lineage 2, which, unlike the earlier literatures that says that lineage 3 is the dominant one, what we saw in our own environment throughout was second lineage. And we recommended that the position of the Nigerian government should not be as if we only have lineage 3 in circulation. We have also another lineage that is circulating in our own environment. Mr. Chairman, this was on PPR. Then come the disease that I am in love with so much, Newcastle disease. In using this disease, Mr. Chairman, I have studied every component of virology. whether it is participatory epidemiology, whether it is field epidemiology, whether it is diagnostic virology, whether it is host and pathogen interactions, or even pathogen genomic surveillance, vaccine development, diagnostics, every aspect of virology on this disease I have studied. Mr. Chairman, the disease can wipe out more than 100%. It can wipe out the whole 100% of birds. I think this is a disease common to everyone. Since we were young, we know how this disease is. It has different names in our own locality. So different people in different language call it different names. So it kills them like crazy. We do vaccination, but still there is a problem. So, Mr. Chairman, and the disease is really distributed worldwide. Nigeria is inclusive. So we now ask this question: what exactly is the problem? Why is it that we are vaccinated, yet we are having problems? We ask, is it really vaccine failure? Or is it that we are not doing vaccination very well? Or is it due to viral evolution, which of course we know is a characteristic of RNA viruses? Or is it really genetic mismatch? So in order to answer this question, Mr. Chairman, I have some of students also. I like calling my students who they are, talented students, Mr. Chairman. Dr. Sadiq and Dr. Amy, they investigated for the very first time, as far as we remember. We isolated the live virus here in Sokoto, Mr. Chairman, against all odds. I mean, despite all the problems of logistics and everything, we were able to isolate the virus. And not only isolate this virus, Mr. Chairman, we were able to characterize it and showed the exact viruses that are circulating in our own environment, Mr. Chairman. Using some sequencing technologies and phylogenetics and bioinformatics, we were able to reveal that in Nigeria, we have many lineages. By the way, Newcastle disease has only one serotype, but several genotypes. And then there is a problem. Most of the vaccines are one genotype, and the circulating strains are another genotype. So we showed that there are multiple genotypes in circulation. You can go to the next slide. First of all, we saw we have the genotype 6, we have genotype 14B. I'm sure Prof. Musa Osman would smile at this. We have genotype 14B, we have genotype 17, genotype 18, and so on and so forth. So there are multiple genotypes, and the basic problem we identified here is that all the circulating genotypes we have in our environment are very far genetically from the vaccines that are available. Whether it is I2, B1, Comoros, Lasota, V4, VGTA, every damn type of vaccine that was used in poultry was genetically different from the circulating viruses. Having known this, Mr. Chairman, during my PhD, I did the needful. In Malaysia, where I did my PhD, Mr. Chairman, it was not this particular genotypes that are circulated. It was another genotype called genotype 7. Genotype 7, Mr. Chairman, is known to be very deadly. In fact, people are saying it is the virus or genotype responsible for the 5th pandemic of Newcastle disease. Now, this genotype, we did genetic engineering. We got the virus, shut down the genes that are responsible for virulence and pathogenicity while maintaining all other properties of the virus. SubhanAllah, we were able to rescue the virus with all the other characteristics except the pathogenicity using the power of a technology known as reverse genetics. Mr. Chairman, using reverse genetics, we manipulated the virus. We checked it, and then having done the genetic manipulation, by the way, it took me 15 months of repeated experiment to rescue a virus with altered phenotype. Now we obtained this, Mr. Chairman, and after obtaining this, we checked all the other parameters: stability, pathogenicity, immunogenicity, protective efficacy. All these were tested, and they were all in line with our earlier hypothesis, and we come up with the vaccine candidate. As I'm speaking to you now, Mr. Chairman, this vaccine has already been evaluated, and we got a patent as a result of this vaccine that is now about to be commercialized by one of the largest pharmaceutical companies in Malaysia. Mr. Chairman, sir, apart from this vaccine, we also tried to test the route of administration. We check the route by checking what is the best route of administration. We found out that eye drop, that is intraocular route of administration for this particular vaccine we developed is the best route, and now we recommended for the company that is about to commercialize it that this route should be adopted as the best route for vaccinating poultry in this. Next slide. This is the patent we obtained after my PhD. Next. So, Mr. Chairman, apart from developing vaccine, we also worked on studying host pathogen interaction. Those of us in immunology, they know what we call natural killer cells. Natural killer cells are known to be cells that tend to destroy viruses. They attack viral infected cells, they attack cancerous cells. So we want to know, why is it that despite the presence of natural killer cells in abundance, this virus is still going in there and causing problem? I mean, this is quite surprising. So we tried to investigate. And what we found also, it was really amazing, Mr. Chairman, because we demonstrated that the virus will instead, this particular cell that is capable of killing the virus even preferentially go inside the cell and replicate. And how does it do it? It goes in and then try to shut down or reduce or downregulate the expression of certain genes that are responsible for the activity of these natural killer cells. This is what we identified in this. Mr. Chairman, apart from NDV, we also tried to expand our lens in avian virology by studying another virus known as IBDV. IBDV is a virus, Mr. Chairman, which is also affecting poultry, that tends to attack poultry immune system. It destroys what we call bursa of fabricius, one of the major immune organs in poultry. And once it destroys it, then the whole problem will begin. Mr. Chairman, using this, first of all, we try to understand, okay, how is this virus capable of evading the innate immunity and cause disease in poultry? We did the same thing we did with Newcastle disease. And unlike NDV, this particular virus we demonstrated that it cannot really enter into natural killer cell, but somehow it surrounds it. We showed it using transmission electron microscopy, that the virus surrounds the cells, but then it is incapable of penetrating. Still, it is capable of remotely reducing the expression of these genes that are responsible for the activity of these natural killer cells. This was published in a very good top journal, Developmental and Comparative Immunology. Apart from that, Mr. Chairman, I also worked on the genetic or molecular diversity of this particular virus, alongside with my friend, the head of Department Veterinary Microbiology, Professor Nafi Ulawal. Mr. Chairman, in this study, we tried to check the diversity of the virus. And then we compared the early passage and low passage and showed that there is difference actually in terms of the accumulation of mutation between the two passages, and now make recommendations that have a lot of implication in vaccine development. Yes, so Mr. Chairman, apart from IBDV, we also worked on IBV, which is also another poultry virus. This particular virus, Mr. Chairman, unlike IBDV and NDV, is known to be also highly genetically diverse. It is also a virus that is capable, it doesn't really kill like NDV, but it is causing a lot of loss of production, especially loss in egg production, which is, to a farmer, a lot of losses economically. So one of the studies we did was with my students also, my MSc student, Dr. Buhari, who is also a student, who is also a staff, by the way, in this university at the moment. We tried to investigate the genetic diversity across Africa. And we found out that in Africa, we have a lot of genotypes of this particular virus, so many variants. And some of these variants are also genetically distant from, the vaccine strains. And we recommended the so-called importance of regionally important vaccines, so that we have to develop vaccines based on the currently circulating genotypes of this virus. Next slide. So these are some of the contributions made, Mr. Chairman, in terms of livestock and avian virology. Now moving to the contributions in human virology. The contributions made in human virology do not represent any deviation from my earlier focus, Mr. Chairman. The science remains the same. It is all about molecular epidemiology, all about vaccine development, vaccine design. It is just the change in host, Mr. Chairman. Because why human virology? Because, Mr. Chairman, 70% of all emerging infectious diseases, they are from animals. They are as a result of zoonotic spillover infections. And zoonotic spillover infections, even the recent outbreak of Ebola, the Ebola outbreak of 2014 to 2015, Mr. Chairman, it was said to be as a result of encroachment by human activities of forest, leading to dismantling of bats that harbor this virus, they came closer to human beings, spilled over the virus, and there was escalation, so that outbreak of this disease kept occurring. If you look at COVID-19, people are still saying that it was as a result of contamination from laboratory, but the virus that was obtained, from a bat. So virtually most of these pandemics that have occurred in the history, they are as a result of zoonotic spillover. That is why I felt there was a need to also increase the impact of what we are doing by not only restricting to health for food security, but global health security for human survival, Mr. Chairman. And hence the title of this lecture, Confronting Viruses with Vaccines from Head Health to Human Survival. Next slide. Previous slide. So the first thing we did, Mr. Chairman, was to identify the most dominant disease that was quite neglected, which is Lassa fever. Lassa fever, every now and then on a weekly basis, we keep seeing a report from NCDC that many people have died of Lassa fever, yet no vaccines. Right from 1968, when this virus started, still no vaccine till to date is licensed, perhaps due to some political reasons. From 1968, this disease has been restricted within the West African confines. It is found only in West Africa. So since it is West African problems, if we do not solve our problem, nobody is coming to solve our problem. So this is the initiative that made us to really apply for a grant from TEDFund. And we are grateful to TEDFund for graciously granting us huge sum of money to develop the first candidate of Lassa fever. And using this particular grant, Mr. Chairman, we have been able to design the vaccine, to construct the vaccine, and not only construct the vaccine, but to validate the expression. As I'm speaking to you, the vaccine is being evaluated in animal trials for immunogenicity and possibly for immune protection. Alongside, Mr. Chairman, the most beautiful part of this story is all these things were done here in Sokoto, so these things are doable here. So we are able to not only do what we can, but also do the training and capacity building for next generation of scientists. The next thing, Mr. Chairman, as we are doing this, then COVID-19 hit. And that is why I still hail the management of Usman Danfodio University. I continue to hail this university, Mr. Chairman. We were the first in this university to alert the whole country of the looming consequences of COVID-19. And this was done through a symposium, the first national symposium on emerging and re-emerging infectious diseases, organized by this same university. So we did this symposium, and I was privileged to have presented alongside the Director General of NCDC and one professor of infectious diseases. Mr. Chairman, I could still remember what the DG NCDC said. He said that, Usman Danfodio University, you are leading this. Meanwhile, this is our job. We are supposed to have done it, yet you take the lead and do it. Thank you very much, Usman Danfodio University, for doing this. Mr. Chairman, following this, COVID came to Nigeria. When COVID came to Nigeria, we formed a team. I was part of the state team that was testing for COVID, and we were the frontliners. I remember how many people were calling me COVID man in those days, because we were the ones frontliners doing COVID testing in this state. This led me to become member of a COVID-19 research consortium set by NCDC, and I was on the advisory committee on COVID-19 vaccines. And later, Mr. Chairman, I was involved with other scientific activities during this COVID-19. We were also involved in capacity building, Mr. Chairman. We formed a vaccine development consortium from 5 different institutions. Usman Danfodio was leading. We have NARIC, we have University of Jos, we have NIMA, we have NBRI. We came together and wrote a joint grant, and we were able to secure a grant that led to the development of the first recombinant DNA vaccine candidate against COVID-19 here in Nigeria. Mr. Chairman, sir, it was not only a COVID response, but we are involved also in research during COVID-19. I remember how our mentor here, Professor BELLO, who was also head of candidate therapeutics at NCDC, NCRC, of which I was also a member in those days, Mr. Chairman, we did some study that combined computational virology and laboratory validation. We were able to do drug repurposing because it was too late to take a very long time to develop a new drug. We used the existing drugs and repurposed them, and we found, he called it magic bullet, because we were able to find that some of the existing drugs that were approved for something else actually had some activity against COVID-19, and we were able to publish that in some reputable journals also. Mr. Chairman, so moving on in COVID-19, we went through a lot of things: national response, diagnostic capacity strengthening, epidemiological research, vaccine development, therapeutics, and of course scientific communication. Mr. Chairman, here comes my favorite, dengue virus. If you go to Saudi Arabia at this moment and go to the scientific community and mention the name BELLO, the next thing they will mention is dengue, because of my work on dengue virus. Mr. Chairman, sir, upon arriving in Saudi Arabia some few years ago, I was looking for a research problem. I realized that dengue fever, unlike what people used to know that it is prevalent in swampy areas, it is actually existing in Saudi Arabia, but nobody ever paid attention to it. It was first reported as early as 1994, but still, not many people were into it. So we tried to investigate, no data at all. We tried to investigate and went to the gene bank, checked, and there was no information. We went to the field, and we were able to identify the virus and conduct I was able to lead the national surveillance, genomic surveillance of this pathogen throughout that period, and we were able to populate the national, NCBI database with new set of genomes of this virus that was published in a very top journal in the field of infectious disease, Journal of Infection, as the first genomic surveillance of dengue virus in the Kingdom of Saudi Arabia. Mr. Chairman, apart from this genomic surveillance, we moved to diagnostics. Diagnostics, we were trying to find out here in Africa and other resource-poor countries that have difficulty in accessing very expensive diagnostics. How can we come up with a cheap but effective diagnostics? My student, Zulaihat, she dealt with that, and we were able to come up with some recommendations of the type of diagnostics that will be appropriate for our own settings here. And we communicated that in one of the Cambridge UK journals, which is considered top journals in the field of virology also. Apart from that, Mr. Chairman, we also worked on innate immunity of this virus. We tried to see, okay, this disease has different phases. We have mild disease, we have severe, we have moderate. We wanted to know, why is the difference occurring? So we checked it from the angle of innate immunity, and we tried to check through my student known as Dawood, Mr. Chairman. We tried to check the expression of certain innate receptor genes, and we analyzed it. We found out that the reason why dengue is more severe compared to other viruses is because it is capable of causing excessive release of some cytokines. So it is not about only the pathogenicity of the virus, it is the type of immune response that it elicits that adds to the pathogenicity and complexity and severity of the disease. And now we recommend that in trying to develop any vaccine, we have to consider the ability to elicit and activate some of these TLR7 and TLR8 receptors. So this is about the innate immunity. And now, Mr. Chairman, my very ongoing work, what is now my identity in Saudi Arabia, which is the development of universal dengue vaccine using a technology known as messenger RNA. Mr. Chairman, sir, dengue vaccine has been approved in 2015, but immediately after it was used, then there was a problem in Philippines. Many people began to die and they began to show some abnormalities. And when they checked, they found out that the problem was actually, the vaccine was effective, but there was a phenomenon known as antibody-dependent enhancement of viral infection that was characterized by this. So the antibodies that are elicited by this particular vaccine they have developed and approved, those antibodies, in addition to being protective, they are also, some of them are sub-neutralizing. The FAB region, those of us who are immunologists will know this. FAB region, they combine it, and the FC region is free. The FC region is capable of communicating with the FC gamma receptors on some of these monocytes, macrophages, and other cells. And this will lead to the granulation and cause a lot of, release of some cytokines and other things that will tend to complicate the disease. So in order to develop a new vaccine, You have to avoid ADE stimulating motif. That is what exactly we are doing in this technology. So instead of using the whole virus as an antigen, we are trying to extract only the unique portions that are devoid of this ADE stimulating motifs, and then we are using that as template. Right now, as I'm talking to you, we have constructed the vaccine, designed the vaccine, we have validated it. As of this afternoon, Mr. Chairman, we are preparing for animal trials to test for the immunogenicity of this tetravalent universal vaccine in Saudi Arabia. So, Mr. Chairman, I have worked on a lot of other viruses, but there is no time for us to go through all these viruses. So I will summarize by saying this, that we have, throughout my career, Mr. Chairman, I was able to secure a lot of research grants. And these research grants were both in Nigeria and internationally, and the Kingdom of Saudi Arabia. In Nigeria, TED Fund was my major funder. TED Fund was able to graciously fund lots of my projects. These are some of the research projects I was the principal investigator. I have quite a lot of them that are too numerous to mention, Mr. Chairman, as co-investigator, funded by different funding agencies. One of them I'm very proud of, Mr. Chairman, is that highly competitive one funded by Saudi NIH, which was just recently obtained. This grant, I will speak about this a little bit later, is about platform development. But these are some of the grants I was able to win throughout my career. So moving through the future, Mr. Chairman, there's one reality we have to face. And this reality is that viruses will never stop emerging. That is the truth, Mr. Chairman. Viruses will continue to emerge, whether we like it or not. I'm very sorry to say this. Pandemics will still occur. So diseases will still occur, but then, Mr. Chairman, all we need to do is to prepare. Pandemic preparedness is the answer. We have to go through pandemic preparedness. And through pandemic preparedness, Mr. Chairman, because viruses will never stop emerging, we have to prepare. And one of the cardinal things I recommend is to embark on genomic surveillance of pathogens. Genomic surveillance of pathogens is now what WHO is doing, and now what many other countries are adopted. We have to emphasize on this. The next thing, Mr. Chairman, we also have to pay attention to is to come up with platforms. Now it's not about developing a single vaccine. What we are trying to do, the grant I just obtained by Saudi NIH, it's not about developing a vaccine against a single disease. No, it's about creating a vaccine platform that will serve as a plug-and-play system, so that if there is the platform in existence, any disease that comes up, all we need is to remove the antigen and copy and paste the next antigen, and we got our vaccines. So this establishment of a flexible virus-like particle vaccine platform for the for the removal and elimination of emerging and re-emerging infectious diseases, that is what I try to advocate, not only in Saudi Arabia, but in Nigeria and in fact in Africa in general. And lastly, Mr. Chairman, I want to advocate for building capacity in Africa. I am so happy, Mr. Chairman, recently, courtesy of COVID-19, we are seeing quite a lot of interest in some of this infectious disease research. I happen to be, part of the stakeholders that are building capacity in the University of Rwanda, East African Center of Excellence on Vaccines, Immunization, and Health Supply Chain Management. They are trying to come up with something for Africa. Africa CDC is trying to set a target. By 2063, we have to be able to produce our own vaccines. Africa is trying to, come of age, because we are the largest consumers of vaccines. 99% of all the vaccines that are used in Nigeria, they are imported vaccines. Meanwhile, we have MVRI, we have NIMA, we have many other. We have quite a lot of brilliant scientists in this country, yet we allow ourselves only to be consumers. The answer is we have to develop our own vaccines. And now, not only develop our own vaccines, Mr. Chairman, we do not have to be like assembly of vaccines centers. No, we have to be capable of developing it from the antigen, from the scratch. We should own our own antigen, then our own development profile. Then we should be able to have an end-to-end training in vaccine development. That is what I'm trying to advocate in this aspect. And finally, Mr. Chairman, on a final note, I want to say this. that from head health to human survival, Mr. Chairman, our greatest defense against emergent viruses lies in transforming knowledge into innovation and innovation into vaccines. Finally, Mr. Vice Chancellor, sir, this has been my story. This has been my science. And today, I have fulfilled my obligation to the university by delivering my inaugural lecture. I thank you all for your kind attention. Terima kasih banyak-banyak. Allah yatikum al-afiyah jami'an. Barakallahu fikum. Assalamu alaikum wa rahmatullah.